Welcome to this tutorial on digestion and assimilation of carbohydrates, brought to you by Medical Biochemistry Power Capsule series. This video covers the following CBME curriculum topic: describe the processes involved in digestion and assimilation of carbohydrates from food. To understand the digestion and assimilation of carbohydrates, it is essential to be familiar with the various types of carbohydrates that enter our digestive system through the food we consume. Carbohydrates entering our digestive system as part of our food can be categorized into three main types: sugars, polysaccharides, and fibers. Each category includes several specific carbohydrates, which vary in their structure and digestion process. Principal monosaccharides include glucose, fructose, and galactose. Glucose is found in fruits, vegetables, honey, and various processed foods. Fructose is present in fruits, vegetables, honey, and high fructose corn syrup used in sweetened beverages and processed foods. Galactose is typically obtained through the digestion of lactose, found in dairy products. The main disaccharides include sucrose, lactose, and maltose. Sucrose or table sugar is present in sugarcane, sugar beets, fruits, vegetables, and many processed foods. Lactose is present in milk and dairy products. Maltose is present in malt products and formed during the digestion of starches in foods like bread and cereals. Oligosaccharides include raffinose, statiose, fructooligosaccharides, and galactooligosaccharides. Raffinose is found in beans, cabbage, Brussels sprouts, broccoli, asparagus, and whole grains. Statiose is present in beans and legumes. Fructooligosaccharides are found in onions, garlic, leeks, asparagus, artichokes, and bananas. Galactooligosaccharides are present in beans, lentils, chickpeas, and other legumes. The dietary polysaccharides include mainly starch and glycogen. Starch is found in foods such as potatoes, rice, wheat, corn, and other grains. Glycogen is not commonly consumed directly, but it can be obtained from animal liver and muscle tissue in meat. It does not constitute any significant portion of carbohydrate that we ingest. Polysaccharides which cannot be digested, such as cellulose, are included under the category of fibers. Here, we have considered only the digestible polysaccharides. Dietary fiber includes soluble and insoluble fiber. Soluble fiber dissolves in water and forms a gel-like substance and is found in oats, barley, nuts, seeds, beans, lentils, peas, and some fruits and vegetables. Insoluble fiber does not dissolve in water and helps add bulk to the stool. It is found in whole grains, nuts, beans, and vegetables like cauliflower, green beans, and potatoes. Inulin is found in chicory root, onions, garlic, and leeks. Lignin is found in the seeds and woody parts of fruits and vegetables. Pectin is found in fruits such as apples, citrus fruits, and berries. Cellulose is found in the cell walls of plants, including vegetables, fruits, and whole grains. Beta-glucan is present in oats and barley. Hemicellulose is present in whole grains and the outer skins of fruits and vegetables. What about the digestibility of carbohydrates? Not all carbohydrates are digestible. Some can be digested, some can be partially digested, and some not at all. Normally, most monosaccharides and disaccharides can be digested. Dietary polysaccharides, such as starch and glycogen, can also be digested. However, most oligosaccharides cannot be digested. But some of these can be fermented by gut bacteria and are therefore useful. By and large, dietary fibers cannot be digested. However, similar to oligosaccharides, some of these can be fermented by gut bacteria. Before we embark on exploring the process of digestion and assimilation of carbohydrates, let's first conduct a succinct review of the destiny of non-digestible carbohydrates. While the majority of carbohydrate digestion and absorption occurs in the small intestine, some carbohydrates reach the large intestine undigested. These include dietary fibers, resistant starches, and certain oligosaccharides that are not broken down by human digestive enzymes. The large intestine, or colon, plays a crucial role in the fermentation of these non-digestible carbohydrates, contributing to gut health and overall well-being. The first step is the arrival of non-digestible carbohydrates to large intestine. Non-digestible carbohydrates that escape digestion in the small intestine enter the large intestine. These carbohydrates are typically composed of complex polysaccharides such as cellulose, hemicellulose, pectins, and resistant starches. These compounds resist enzymatic breakdown in the upper gastrointestinal tract due to their structure or bonding, microbial fermentation, production of short-chain fatty acids, and gas production are three important aspects related to the processing of carbohydrate in the large intestine. Oligosaccharides, such as raffinose, statiose, fructooligosaccharides, and galactooligosaccharides, are short chains of monosaccharide units. Human digestive enzymes cannot fully break down some of these oligosaccharides. These oligosaccharides are not digested by human enzymes in the small intestine because we lack the enzyme alpha-galactosidase, which is necessary to break the bonds between the galactose units and the other sugars. When raffinose and statiose reach the large intestine, they are fermented by gut bacteria, producing gases like hydrogen, methane, and carbon dioxide, as well as short-chain fatty acids. The large intestine is densely populated with a diverse community of microorganisms, collectively known as the gut microbiota. These microbes possess enzymes that humans lack, enabling them to ferment non-digestible carbohydrates. The fermentation process involves the breakdown of these carbohydrates into smaller molecules through microbial enzymatic activity. The fermentation of oligosaccharides in the large intestine contributes significantly to gut health by supporting a healthy microbiome and producing metabolites that can be absorbed and used by the body. Fructooligosaccharides are chains of fructose molecules, often ending in a glucose molecule. Fructooligosaccharides cannot be digested by human enzymes in the small intestine. Instead, these substances pass into the large intestine where they are fermented by beneficial gut bacteria. The fermentation of fructooligosaccharides promotes the growth of beneficial bacteria, such as bifidobacteria and lactobacilli, contributing to gut health and the production of short-chain fatty acids. In the large intestine, bacterial enzymes like fructan hydrolase and other fructosidases break down fructooligosaccharides. Galactooligosaccharides are chains of galactose units ending in a glucose molecule. Like fructooligosaccharides, galactooligosaccharides cannot be digested by human enzymes in the small intestine. Galactooligosaccharides reach the large intestine intact, where they undergo fermentation by gut bacteria. Galactooligosaccharides act as a prebiotic, fostering the growth of beneficial gut bacteria, which in turn produce short-chain fatty acids that can be absorbed and utilized by the body, similar to fructooligosaccharides. Bacterial enzymes in the large intestine, such as beta-galactosidase, help break down galactooligosaccharides. The large intestine plays an important role in maintaining health and well-being. One of the primary outcomes of microbial fermentation is the production of short-chain fatty acids, including acetate, propionate, and butyrate. These short-chain fatty acids are produced as end products of the fermentation process and have several important functions. Short-chain fatty acids serve as an energy source for colonocytes and can be absorbed into the bloodstream to be used by the body. Butyrate is particularly important for colonocyte health and function. Short-chain fatty acids help lower the pH of the colon, creating an environment that inhibits the growth of pathogenic bacteria and promotes the growth of beneficial bacteria. Short-chain fatty acids have been shown to have anti-inflammatory properties, improve gut barrier function, and influence metabolic processes, such as lipid metabolism and glucose homeostasis. The fermentation process and the resulting production of short-chain fatty acids contribute to maintaining a healthy gut microbiome. A balanced microbiome supports digestive health, enhances immune function, and protects against pathogens. Moreover, the presence of dietary fibers and resistant starches can increase stool bulk and promote regular bowel movements, aiding in the prevention of constipation and other gastrointestinal disorders. What are prebiotics, probiotics, and synbiotics? Non-digestible carbohydrates that promote the growth of beneficial bacteria in the gut are known as prebiotics. Common prebiotics include inulin, fructooligosaccharides, and galactooligosaccharides. Probiotics are life-beneficial bacteria, and symbiotics combine prebiotics and probiotics. Consuming prebiotic-rich foods supports a healthy microbiome. Additionally, symbiotics, which combine prebiotics and probiotics, can further enhance gut health by providing both the required microbes, as well as the substrates they thrive on. Through the action of all these, we can basically have a thriving and happy microbiome. A byproduct of fermentation is the production of gases, including hydrogen, methane, and carbon dioxide. While these gases are typically passed harmlessly, excessive gas production can lead to bloating and discomfort in some individuals. The composition and activity of the gut microbiota, as well as the type and amount of non-digestible carbohydrates consumed, influence the extent of gas production. The fermentation of non-digestible carbohydrates in the large intestine highlights the importance of dietary fiber and complex carbohydrates in the diet. These processes not only contribute to digestive health, but also have systemic benefits, including improved metabolic health and reduced risk of chronic diseases such as obesity, type 2 diabetes, and colorectal cancer. In summary, the large intestine's fermentation of non-digestible carbohydrates plays a vital role in maintaining gut health and overall well-being. By producing beneficial short-chain fatty acids and supporting a healthy microbiome, this process underscores the importance of including dietary fibers and complex carbohydrates in the diet for optimal digestive and systemic health. In the subsequent section, we shall delve into the digestion and assimilation of digestible carbohydrates. Theoretically, these 8 different carbohydrates shown here comprise the major digestible fraction in our diets: glucose, fructose, galactose, sucrose, lactose, maltose, starch and glycogen. However, it depends on our diet. Obviously, those who consume more fruits will have plenty of fructose in their diet, while those who consume dairy products will be consuming plenty of lactose. Galactose and maltose do not usually enter through our food, but are rather formed as products of hydrolysis of starch and lactose. Glycogen is negligible in our diets. One can say that starch is the most important carbohydrate component in our diets. It forms the bulk. As for glucose, we hardly consume it as such, except under specific circumstances. The glucose that enters our system comes mainly from hydrolysis of starch. And those who aren't too fond of sweets have very little amount of sucrose going into their system. Carbohydrates are a primary source of energy for the body, found in foods like bread, rice, fruits, and vegetables. The digestion of carbohydrates involves their breakdown into simple sugars, primarily glucose, which can be absorbed into the bloodstream and utilized by cells for energy. This process involves a series of coordinated events that start in the mouth and continue through the stomach, small intestine, and large intestine. The major final products of carbohydrate digestion are glucose, fructose, and galactose. The digestion of carbohydrates begins in the mouth, where mechanical and chemical processes take place. Chewing breaks down food into smaller pieces, increasing the surface area for enzymes to act upon. Salivary amylase starts the chemical breakdown of starches into maltose, isomaltose, etc. Although this enzymatic activity is brief, as food spends a short time in the mouth, it marks the first step in carbohydrate digestion. Once the chewed food, now called bolus, is swallowed, it travels down the esophagus and into the stomach. In the acidic environment of the stomach, the activity of salivary amylase is halted, as it is not stable in low pH. There is minimal carbohydrate digestion in the stomach. The primary function here is to churn the food and mix it with gastric juices, creating a semi-liquid mixture called chyme, which then moves to the small intestine. Digestion is required only for starch and disaccharides. The monosaccharides in our food are not required to be digested, they can be absorbed as such. In fact, starch and disaccharides have to be converted to monosaccharides in order to be assimilated. The majority of carbohydrate digestion occurs in the small intestine. As chyme enters the duodenum, it is mixed with pancreatic juice, which contains pancreatic amylase, an enzyme that continues the breakdown of starches into maltose and other disaccharides. The small intestine's lining also produces enzymes, such as maltase, sucrase, and lactase, which further hydrolyze disaccharides into monosaccharides, including glucose, fructose, and galactose. These simple sugars are then absorbed into the intestinal cells through specific transporters and enter the bloodstream. Salivary amylase in the mouth converts starch to maltose and isomaltose, which are disaccharides. The pancreatic amylase too does the same. The intestinal digestive enzymes finally break down the disaccharides that come along with the stomach contents. Maltose, isomaltose, sucrose and lactose are broken down by maltase, isomaltase, sucrase and lactase, and the mixture now contains glucose, fructose and galactose as the final products, which are ultimately absorbed. Disaccharides cannot be absorbed, only monosaccharides can be absorbed. The small intestine is the primary site for the digestion and absorption of carbohydrates, with its intricate structure and specialized functions, making it highly efficient for this purpose. The various steps can be enumerated as follows: arrival of chyme in the duodenum, action of pancreatic amylase, action of brush border enzymes, absorption of monosaccharides, and transportation to liver. When chyme, the semi-liquid mixture of partially digested food and stomach juices, enters the duodenum, which is the first part of the small intestine, it triggers the release of pancreatic juice. This juice contains digestive enzymes and bicarbonate ions that neutralize the acidic chyme, creating a suitable environment for enzymatic activity. The small intestine's ability to digest and absorb carbohydrates is coordinated with other digestive processes through hormonal signals. For example, the hormone cholecystokinin stimulates the release of pancreatic enzymes, and secretin prompts the pancreas to secrete bicarbonate-rich fluids to neutralize stomach acid in the chyme. This coordinated process ensures that carbohydrates are efficiently broken down into their absorbable units and properly utilized by the body, providing a steady supply of energy and maintaining metabolic balance. Pancreatic amylase, an enzyme produced by the pancreas, continues the breakdown of complex carbohydrates into simpler sugars. Specifically, it targets starch and glycogen, breaking them down into maltose, maltotriose, and small branched carbohydrates called dextrins. The inner lining of the small intestine is covered with villi and microvilli, forming the brush border. These structures vastly increase the surface area for digestion and absorption. Embedded in the microvilli membrane are brush border enzymes, including maltase, sucrase and lactase, which play crucial roles in carbohydrate digestion. Maltase breaks down maltose into two glucose molecules. Sucrase splits sucrose into glucose and fructose. And lactase hydrolyzes lactose into glucose and galactose. The final products of carbohydrate digestion are monosaccharides, glucose, fructose, and galactose, and these are absorbed by the enterocytes through specific transport mechanisms. There are two main types of transporters, SGLT1 and GLUT5. Another glucose transporter, GLUT2, is also involved. SGLT is abbreviation for sodium-dependent glucose transporter. GLUT is the acronym for glucose transporter. Glucose is the major monosaccharide, which is absorbed from the intestinal lumen into the bloodstream through intestinal cells. There are specific transport proteins involved. Please follow this animation. The red balls represent sodium ions. The blue squares represent glucose molecules, and yellow hexagons represent potassium ions. The sodium-potassium ATPase pump, which is located on the basolateral aspect of intestinal cell, pumps 3 sodium ions out of the intestinal cell for every 2 potassium ions pumped into the cell. This is similar to the action of sodium-potassium ATPase elsewhere in the body. This process creates a low concentration of sodium inside the cells. As a result, sodium ions flow through another transporter known as sodium-dependent glucose transporter, which is located on the apical aspect of the enterocytes. When sodium ions enter the intestinal cells, glucose molecules to enter. Hence, it is sodium-glucose cotransport. For every molecule of glucose, typically 2 molecules of sodium are required to be transported. Once inside the intestinal epithelial cell, glucose then flows through another transporter known as GLUT2, which is located on the basolateral aspect of the cell as shown here. By flowing passively through GLUT2, glucose enters the bloodstream. In this way, glucose is absorbed from the intestinal lumen into the bloodstream, and energy is indirectly required for this process because the sodium pump uses energy derived from the hydrolysis of ATP. Absorption of galactose is also by the same process. To summarize, in the intestinal epithelium, secondary active transport by sodium-glucose transport protein 1 takes place. SGLT1 uses the energy in a downhill transmembrane movement of sodium ions to transport glucose across the apical membrane against an uphill glucose gradient so that the glucose can be transported into the bloodstream. Congenital defects in this transport protein can cause defective absorption of glucose and galactose. GLUT5 or glucose transporter 5 is responsible for the facilitated diffusion of fructose into the anorecytes. Once inside the anorecytes, these monosaccharides are transported across the cell and released into the bloodstream via the transporter, GLUT2. On the basolateral side of the cells, GLUT5 is also found in testes and kidneys. Its sole function is to transport fructose. Its ability to transport glucose is negligible. On the other hand, GLUT2 is a major glucose transporter, which is also found in liver cells, beta cells of pancreas and in kidneys. The transport through GLUT5 and GLUT2 is passive facilitated diffusion. It's not active transport. GLUT2 transports glucose as well as fructose from the intestinal cells to bloodstream. Glucose transport mechanism has already been discussed. GLUT5 is mainly devoted to fructose transport. After monosaccharides have been absorbed from the intestine, they enter the hepatic portal vein and are transported to the liver. The fate of these monosaccharides in the liver depends on their chemical nature and the body's metabolic needs. Glucose mainly enters into glycolysis, glycogenesis, or HMP shunt pathway. Glucose can be phosphorylated to glucose 6-phosphate and enter the glycolysis pathway to produce pyruvate, which can then be used for energy production via the citric acid cycle, or can be converted into lactate under anaerobic conditions. In conditions where glucose is abundant, such as after a meal, glucose 6-phosphate can be converted to glycogen for storage in the liver. This process is called glycogenesis. Some glucose 6-phosphate can also enter the pentose phosphate pathway to produce an ADPH and ribose 5-phosphate, which are essential for fatty acid synthesis and nucleotide synthesis, respectively. Fructose is primarily phosphorylated by fructokinase to fructose 1-phosphate. Fructose 1-phosphate is then split by aldolase B into dihydroxyacetone phosphate and glyceraldehyde. Dihydroxyacetone phosphate then enter glycolysis or gluconeogenesis pathways. Glyceraldehyde can be phosphorylated to glyceraldehyde 3-phosphate and enter the glycolysis pathway, or can be converted to glycerol 3-phosphate for triglyceride synthesis. Galactose is phosphorylated by galactokinase to galactose 1-phosphate. Galactose 1-phosphate is then converted to glucose 1-phosphate. Glucose 1-phosphate can be converted to glucose 6-phosphate, which can then enter glycolysis or be used for glycogen synthesis. Mannose, a minor monosaccharide, is phosphorylated to mannose 6-phosphate by hexokinase. Mannose 6-phosphate can be isomerized to fructose 6-phosphate by phosphomannose isomerase. Fructose 6-phosphate then enters the glycolysis pathway, leading to energy production. Let's try to understand the basics of some common disorders related to digestion of carbohydrates. Several disorders can affect the digestion and absorption of carbohydrates, leading to a variety of gastrointestinal symptoms and overall health implications. These disorders often result from enzyme deficiencies, immune responses, or structural issues within the digestive system. The noteworthy ones are as follows: lactose intolerance, celiac disease, irritable bowel syndrome, fructose malabsorption, SIBO. which is the acronym for small intestinal bacterial overgrowth and pancreatic insufficiency. Lactose intolerance is a common digestive disorder resulting from a deficiency in lactase, which is the enzyme responsible for breaking down lactose. Lactose is the sugar found in milk and dairy products. This condition affects a significant portion of the global population, with varying prevalence across different ethnic groups. In individuals with lactose intolerance, undigested lactose passes into the large intestine, where it is fermented by bacteria, producing gases and short-chain fatty acids. This fermentation process can cause symptoms such as bloating, gas, diarrhea, and abdominal pain. Management of lactose intolerance typically involves dietary modifications to limit or avoid lactose-containing foods. Lactase enzyme supplements can also be taken before consuming dairy products to aid in lactose digestion. Additionally, lactose-free dairy products and alternative milk sources can be used. Celiac disease is an autoimmune disorder triggered by the ingestion of gluten, a protein found in wheat, barley, and rye. It affects approximately 1% of the global population. In individuals with celiac disease, the immune system mistakenly targets and damages the villi in the small intestine upon exposure to gluten. This damage impairs nutrient absorption, including carbohydrates, leading to malnutrition and various gastrointestinal and systemic symptoms. Though celiac disease is not directly related to carbohydrates, but rather related to proteins, yet it deserves mention because wheat is a common source of starch and has a significant role in global food security. Common symptoms of celiac disease include diarrhea, weight loss, abdominal pain, and bloating. Extraintestinal symptoms can include anemia, osteoporosis, and neurological issues. Diagnosis is typically confirmed through serological tests for specific antibodies and a biopsy of the small intestine. The primary treatment for celiac disease is a strict, lifelong gluten-free diet. Avoiding all sources of gluten helps heal the intestinal lining and prevents symptoms and complications. Irritable bowel syndrome is a functional gastrointestinal disorder characterized by symptoms such as abdominal pain, bloating, and altered bowel habits such as diarrhea, constipation, or both. IBS affects a significant proportion of the population, particularly young and middle-aged adults. Many individuals with IBS experience carbohydrate malabsorption, particularly of fermentable oligosaccharides, disaccharides, monosaccharides, and polyols. These poorly absorbed carbohydrates can be fermented by gut bacteria, exacerbating symptoms. Dietary management often involves following a diet regime low in fermentable oligosaccharides, disaccharides, monosaccharides, and polyols. This regime ensures reduced intake of specific carbohydrates known to trigger symptoms. This diet can significantly alleviate symptoms for many individuals with irritable bowel syndrome. Fructose malabsorption, also known as dietary fructose intolerance, occurs when the small intestine absorbs fructose inefficiently. Excessive fructose reaches the colon, where it is fermented by bacteria, leading to gas, bloating, and diarrhea. Symptoms include abdominal discomfort, bloating, gas, and diarrhea, often after consuming foods high in fructose. Diagnosis is usually made through dietary assessment and hydrogen breath tests. Treatment involves reducing the intake of high fructose foods, such as certain fruits, honey, high fructose corn syrup, and some vegetables. Small intestinal bacterial overgrowth is a condition where excessive bacteria populate the small intestine, leading to improper digestion and absorption of nutrients, including carbohydrates. This overgrowth disrupts normal digestive processes, resulting in fermentation of carbohydrates within the small intestine. Symptoms include bloating, gas, diarrhea, and abdominal pain. Small intestinal bacterial overgrowth is diagnosed through breath tests that measure hydrogen and methane levels after ingesting specific sugars. Treatment typically involves antibiotics to reduce bacterial overgrowth, dietary modifications to limit fermentable carbohydrates, and sometimes probiotics to restore a healthy gut microbiome. Pancreatic insufficiency occurs when the pancreas fails to produce enough digestive enzymes, including amylase, which is crucial for carbohydrate digestion. Conditions such as chronic pancreatitis, cystic fibrosis, and pancreatic cancer can lead to this insufficiency. Symptoms include steatorrhea, weight loss, and malnutrition due to impaired digestion and absorption of nutrients. Diagnosis is confirmed through tests measuring fecal elastase levels and imaging studies of the pancreas. Management involves pancreatic enzyme replacement therapy to provide the necessary enzymes for digestion, along with dietary adjustments to manage symptoms and support nutrient absorption. In summary, the digestion and assimilation of carbohydrates are fundamental processes that sustain our bodies by providing essential energy, beginning in the mouth and progressing through the stomach and intestines. Complex carbohydrates are meticulously broken down into monosaccharides. These simple sugars are absorbed into the bloodstream and transported to the liver, where they undergo metabolism. Monosaccharides also enter other organs and undergo metabolism. These processes will be described in subsequent tutorials pertaining to carbohydrate metabolism. The incredible efficiency of our digestive system and the importance of balanced carbohydrate intake for optimal health should now be obvious.
Thanks for watching this video. Very soon, we shall be back with our next tutorial pertaining to carbohydrates, in this Medical Biochemistry Power Capsule series. The tutorials presented in the MBPC series are not intended to be a substitute for your textbooks. However, we feel that these tutorials can prove to be very valuable as complementary educational aids and for purpose of rapid revision prior to exams. If you have enjoyed this video, don't forget to hit the like button, subscribe for more exciting content, and share with your friends, and do keep watching our channel to remain ahead in the game.